Hi everyone, Tam here from Wildlife Queensland. We have results back for the Wolston eDNA sampling that we did back in September, which is really exciting. I'd like to acknowledge the traditional owners upon the land in which we conducted this research in the Wolston Catchment and recognise their continuing connection to lands, water and culture, and pay my respects to elders past, present and emerging. Let's recap on eDNAs for those people who potentially weren't at the workshop as well. So eDNA is the fact that we can get environmental DNA, so fragments of their DNA of species in the environment. They're shedding their hair cells, skin cells, potentially excreting in the water, and we're picking up that DNA in the environment that they live in. It can be aquatic species. We can also get some terrestrial species that live around those waterways and or potentially are passing through as well. It's pretty amazing. It can also be used for soil and sediment, other sources of material in the environment to certainly be able to analyse for, targeted species or multi-species that you may be wanting to develop a program for. For us, it has revolutionised the way we can monitor certainly cryptic species, especially the platypus. We've been doing a lot of that within the Greater Brisbane region for the last 10 years, but certainly mapping potentially invasive species and all those threatened and endangered species as well. It's great because it's non-invasive. We don't actually have to see the animal or harass the animal at all to be able to detect their DNA. And we can either do targeted analysis, so specifically on a species like the platypus, and or a multi-species, so full biodiversity assessment, which is what we excitingly have done for the Wolston Catchment, getting the vertebrate and invertebrate data. Obviously, there are limitations. Like any method, they're not always 100%, and we're not going to be able to detect number of individuals in a system. Unfortunately, it hasn't developed and refined enough in that yet. We can't tell the relatedness between individuals either, or the precise location of the animals. As you can imagine, in a water system and it's flowing, we're getting that movement of the DNA and mixing in the water column. And so the site that you take a water sample from, potentially the DNA has flowed down from upstream, potentially from 500 metres, maybe a bit on even more than that, up to a kilometre. But that also depends on the environmental influences that are breaking down the DNA in the system, because it is still a snapshot in time. So temperature, UV, flow, all of those things will break down that DNA. It's not persistent in the system, so it's still that snapshot in time as to what species we're also capturing. Also, we may also get false negatives. So as much as eDNA is quite sensitive to detect the species in our system, uh, we may not be detecting some of those, especially smaller species like the water bugs and the microorganisms, because we're not getting enough DNA to be able to amplify at analysis stage. So there's limitations in terms of, we're not potentially getting everything that's possibly in this system, but it's a great understanding of a baseline and building that up and understanding potentially what we are and aren't missing using other traditional methods and data and knowledge throughout the community. We can use this as just a very coarse presence absence for species distribution, which is always great. Understanding where things are, we can certainly then focus in on those areas for protection. We can certainly understand if we're doing multiple times throughout the year, different seasonal changes in species abundance and diversity, and we can then develop some occupancy models and abundance models once we're kind of building up that data. We can then bring in other different things to develop those models, so habitat and environmental variables, to certainly understand suitability of habitat for species and things like that as well. So there's a lot of application for the data to be used beyond for understanding our more connected ecosystems. So back on the 29th of September, we had a workshop at the Pooh Corner Environment Centre, and we had a great turnout. It was wonderful to have so many people on board. We learnt a bit about the eDNA, which was wonderful, and then the people that were keen got the eDNA kits and were set off across the 12 sites that we had selected across Sandy Creek, Bullock Head Creek and one up Ricknatras Creek as well. So a big thank you to those who joined us to be able to do that. It was wonderful to certainly be able to get people out and about and actually doing the water sampling. So then those samples came back to the Environment Centre and I sent them off to the eDNA lab, EnviroDNA, down in Melbourne, and analysed for vertebrates and invertebrates, which is a wealth of knowledge and excitement.
To be able to
So this is some photos from they were in the centre. We had a good turnout, and this is one of the Omega Chemical guys, Grant. He's taking a water sample along the kind of bottom edge of Bullock Head Creek. We had the team out, which was really great. So what the results? So the results went back to the lab. It processes those tiny bits of DNA that we're picking up. It amplifies them in the system. There's a data bank, gene bank, that has a lot of the species that are already what they call kind of barcoded to potentially the species level. And that's what we're kind of using to then amplify our results. And so we get kind of a list of species with the number of reads of DNA that it's getting. So it can be as little as in the tens, it can be up to in the thousands. We had over 180 taxa were identified, down to 46 specific species that were identified. We had from native fish, we had a frog, not specifically identified down to a species list. We certainly had some birds, like I said, water birds as well as terrestrial birds. The macropods, there's plenty around, so it doesn't surprise me that we have runoff of their poo and or they're moving in the creeks as well. We see them cross the creeks quite often. Possums, turtles, obviously platypus at sites, ricali, which was great. But then we also detected introduced species, fish, cane toads, and then we've got some certainly domesticated and or potentially wild pigs, dogs within the area as well. And then the invertebrates were incredible. Obviously a lot of invertebrates, and the most that we saw were in the Diptera order, which is the midges and flies, and they're a species that are definitely more tolerant to those pollutant areas. And then we had certain other species as well across a few of the different orders as well, Trichoptera and Ephemoptera and things like that, mayfly, caddisfly, and things. So it was great to see we got a quite an interesting spread across. Now, the exciting thing for you guys is that you can actually go onto this interactive map. So you can scan the QR code and it will load into your tablet or application, and you'll be able to go to this spatial view, and then you can see the sites that we've done, and it gives you the larger size of the actual circle is how much we've found in those areas. But you can click on that site and then it will bring up the species list that we've actually found, right through from the micro or macro invertebrates that we found. So, and you can also see the vertebrates and everything. So you can have a look yourself. You can select by site, so you can go to the site that you actually sampled, and or you can look at species. So say you're wanting to see, I've selected dog here, where the dog sites were, and there's tiny little purple dots that come up on the map, and that's where we had detected dogs. There is a local wild dog, can't remember his name, but he certainly roams around the system as well, so it could be his DNA. Then there's obviously things like the platypus. You can select and see where we actually detected the platypus up on Ricknatras, and then down at the Bullock Head Creek area as well, under the Spine Street Bridge and just downstream of that as well. We know that they certainly move through that system, especially when it is connected and everything. You can then also, continue to explore the other tabs. It gives you a full list of what was detected, and then also some kind of compositions of the different groups that were found as well. So you can kind of play around with a lot of that and see what's there, what takes your interest. If you are interested in more of the insects or anything, you can certainly kind of then delve further into and, select and unselect and work through, if it's mollusks you're into, it'll give you an understanding of kind of what they found and things like that, in a more of that composition kind of style. So yeah, so it's a great interactive map. So please go and have a look at that and see what we found. It's all pretty exciting. So this is just very much a summary of some of the data that we got, and the X's are just representing the presence of the species that were found. The raw data that we get from the Excel spreadsheet would have numbers against those, and also the fact that we did 2 filters per site as well. So I've just combined that in terms of the presence, just for more simplicity of this as well. But if you look at the raw data, which you'll have access to, you can certainly see that there's the reads from tens to thousands in terms of the species that we found. That can potentially, again, it can't tell us numbers. It's not relating to numbers of species, but potentially there's more than one contributing to the DNA if it's quite a high read within that site and things like that. But again, it can be still quite tricky to interpret and understand that DNA. And this is just the start of the program. So potentially we will have a bit more of a handle once we finish up the 5 years of this project. So you can see here it was really great. We detected some of the flying foxes in the system, and obviously platypus and ricali, and certainly the species of different birds and things like that. So it gives you a bit of an idea of the spread of things that we see. From beetle larvae, caddisfly larvae, which are really cool. We had detections of freshwater catfish, striped gudgeons, short-finned eels. As much as people potentially don't like eels, I think they're really quite pretty. I think they were in the list of underrated species for this year. They didn't win. It was the ricali that won, which was pretty exciting. But I think, again, they get a bit of a bad rap because they look, I don't know, maybe a bit scary or something. But really great to see that they were across quite a number of sites, as well as the long-finned eel as well. Saw-shell turtles also came in as well, and the bull routes, which is really cool. So they're a stonefish, a venomous. They have venomous kind of fins that they will spike out and inject. And they certainly come in from the Brisbane River and move up the system when it's fairly well connected with water and things. So we had those at a couple of sites as well. So just kind of delving a bit more into water bugs and the importance of water bugs, or macroinvertebrates in the system. They are a food source for many of our aquatic species, but it's really interesting, and it blows my mind that people have delved so much into the macroinvertebrate world and understanding how sensitive they are, different species, and how sensitive they are to pollution in our waterways. And so they're sensitive to those impacts and disturbances, which is incredible. So we can potentially determine a bit of a habitat quality of our waterways because of the macroinvertebrates in our system. And so you can kind of get, account of the presence and number of the species. So originally you would go out and, the traditional method is sweep netting and getting those live samples to pick out and identify down to species and things. And then you can calculate what is called a signal score, a stream invertebrate grade number average level. And that will then give you a site score as to, I guess, the health of that waterway. And so it's quite amazing that we can do this and then get an understanding because of the diversity and abundance of the different orders of our water bugs in the system, which is really great. So the main orders that we mostly get in our systems, but there's quite a number more than this. You've got your mosquitoes in the Diptera, dragonflies, mayflies, caddisflies, the crustaceans, and then the true bugs as well. So they're the ones that we kind of focus on, but it's really interesting. You can see a gradient as, we potentially move from fairly good habitat, cleaner waters, not the pollution running in. You get certainly the more sensitive species to pollution. and then it kind of goes down. So number 10 is sensitive, and then you're kind of moving down to number 1, and they're tolerant species. So your tolerant species being those mosquito larvae and bloodworms, flatworms, and things like that that we potentially get. So you kind of get a landscape gradient across the system, potentially in terms of the pollution in the area. It's amazing. I think people are still so amazed at what we can find in our waterways and what actually then turns into those flying species around our waterways. So caddisflies, these little ones that make cases, little sleeping bags for themselves with leaf matter or little bits of sand or sticks, or they fit themselves into the sticks, as you can see in the screen there. They turn into the caddisflies, and they fly around everywhere, and then again become food for then potentially other species like our microbats and our birds and things. Dragonfly larvae, I think they're really cool. Our beautiful dragonflies. Then you've got the mayflies with the 3-pronged little tail there, and then they turn into these beautiful, elegant looking little insects as well. Beetle larvae, the little water pennies that they're called, turn into the beetles. And then obviously those kind of smaller wriggly worm kind of things are then, your midges, mosquitoes, flies, and things like that as well. So, but the world of water bugs is incredible. There is so many more than just what we're even seeing today. So it's quite amazing, the species that we can actually pick up. Now, we kind of did a rough identification from our species that we were detected of the water bugs in our data, and we roughly calculated a bit of a signal score from this. And unfortunately, it's not looking too good under the severe pollution. Probably not surprising. I'm sure all of you understand that, especially that came to the workshop. The sites that we went to, highly industrialized, urbanized. There's a lot of stormwater runoff. There's a lot of most likely chemical runoff and all that kind of stuff. And so that is what is influencing the dynamics of the water bugs that are in our system. So it's looking pretty grim at the 2s pretty much across that whole area. However, as mentioned, there is limitations to obviously eDNA. We're not potentially picking up other species of sensitive species because they're probably only a small number of them, so potentially we're not picking those up. Whereas if we're doing traditional methods like the sweep netting and things, we'll possibly get more of that dynamic and representation across the system as well. So yes, there's always kind of limits and caveats to how we interpret our data, but I just thought it would be interesting to see, if we were to apply the signal score to the eDNA data, what we potentially would get. But it also makes sense because, we had 50 species of the Diptera order, and that order is, the pollution sensitivity of their signal score ranges from like 1 to maybe 3 or 4 or something. So we're still in that very lowest scored species regardless. That was the abundance that we saw. So we may not be too far off, but obviously, yes, there has been comparative studies between eDNA and those traditional methods, and they can be comparable, depending on, again, your survey design and what you're trying to achieve and things like that. But that's a full nother, presentation and workshop. So, but in general, it gives us an understanding and a snapshot. But again, it does make sense because it is such a highly impacted waterway, and quite sandy. Yes, we've got Pooh Corner, which is lovely to have that, respite and reserve for wildlife and things. But then look at the rest of the riparian zone. It's quite narrow through the systems. A lot of impacts, a lot of flow, like I said, stormwater runoff and things like that are coming in from everywhere. So there's a lot of impacts to this system, but incredible. We still have species holding on, like platypus, ricali, our turtles. It's amazing to see because they will still eat lower order water bugs. So even though the water bugs may not be diverse in their orders, they're still prolific in what is there, and that is still food for fish and turtles and frogs and things like that. So we're still seeing that full kind of spread of the ecosystem here, which is great. But obviously it needs a bit of love. And so this eDNA can certainly be used to, we give this data to councils, we work with obviously the Wolston and Centenary Catchment, and we can then, put this information out there and say, look, here's platypus, here's ricali, here's the turtles. These are the areas potentially where we need some more targeted conservation actions. If there are invasive species, which unfortunately, yes, what potentially could be, species removal. We can track, monitor, and over time the ecosystem health, and hopefully we will see that within the 5 years of this program, understanding if there are changes across the system over the 5 years that we will be running this program, which will be interesting. Again, a lot of pressures in that system, so it'll be interesting if we do see changes or not see changes, and what that means for the system, again, moving forward in terms of resilience and future for species, especially like platypus, ricali, and things like that. We can certainly, those sites where we do find platypus or other species can highlight areas of value and protection. And that has already been done because we've done eDNA in this system for quite a number of years. And because of this, I think we've been able to then build upon that, and WAC has been amazing in terms of having platypus planting days, and that's supported by council, and Habitat Brisbane from council do a lot. So I think it just gives even more motivation and drive to then focus on these areas, because it's like, oh my gosh, we have an iconic species in this system. So that's really exciting, and we're seeing that, which is really great. And obviously it engages the community. The people that came out for the workshop, amazing. Thank you that you were so interested and you wanted to go out and do the water sampling. I can only encourage you to continue to come along every year. This is going to happen for the next 4 years, to build up that database for 5 years worth of data. But also the WAC group have other amazing community days, platypus planting days, supporting that riparian zone, which is not only important for platypus, but for a lot of species and just the health of our waterways. Obviously, there is a lot more to do connecting with businesses, and we're lucky enough that we've connected with Omega Chemicals, and hopefully through their kind of influence and connections, we can start to have potentially more of those conversations with the local groups and things that are around that area, and bring in more stewardship from the businesses. That would be really great to see. But ultimately, our community is driving a lot of this. So we thank you for your engagement and your support, because ultimately, if we didn't have you, these programs wouldn't happen. Well, they would, but they wouldn't have the impact. They wouldn't have the drive behind them to be able to do it. So we appreciate that. So it's really exciting to see, and a great result. Really interesting data. I'm excited to continue this for the next 4 years. Again, thank you to the volunteers and specifically the bunch of Omega staff that came out. It was great to have you on board to see, what's right in your local backyard and where you're working. If you do have any questions, please reach out to me at platypus@wildlife.org.au. There is also a document that is sent out as well with this, to give you kind of a bit more of information and things about the program and things like that. So have a look at that, and I'll certainly be in touch for round 2 next year, and hopefully see you all back then, and even more new faces. So thank you, and we'll see you in 2026.